So it is my pleasure today to introduce Matthias Rillig, who has been here on campus for the past 2 months at least, I think. So you can often find him out in front of the cafeteria chatting away with people. Matthias came from the Free University of Berlin, where he leads possibly the largest research group I've ever seen of any ecologist specifically. It's like 30 or so odd people at the university's Institute of Biology. He's also the director of the Berlin-Brandenburg Institute of Advanced Biodiversity Research. And before moving to Berlin in the early aughts, he was also a professor at the University of Montana in the United States. His research is quite broad, but encompasses themes in soil biodiversity, soil conservation, microbial ecology, and the impacts of global change on all of those aforementioned things. He's really advanced our understanding of the field of soil ecology and soil conservation, and has made, I would argue, great strides in sort of bringing this to the forefront, because often, when I read like the New York Times or some of the large periodicals nowadays, most of the topics that I see covered relating to ecology or fields close to me often now have to do with soil microbiology. And so, some of Matthias's work has really kind of coalesced a lot of the thinking and societal understanding of the importance of soils, which kind of, go forgotten maybe, or have been a little bit. He's received numerous honors, including election to the German National Academy of Sciences and Academia Europaea. And beyond these scholarly contributions, he also has a great Substack, sort of like the new way we call a blog, and as well as a great YouTube channel where he offers valuable advice for early career researchers. I just read, I think, one recently published on how to avoid a toxic lab situation and why you might want to avoid that. And I found it to be really nice to read and well-reasoned. So please join me in thanking Matthias, who will be talking about experimentally dealing with the impacts of an increasing number of factors, global change and soil as case studies. Thanks.
Well, thanks very much. That's probably the nicest introduction ever, and I think it's going to be downhill from now. But this is a slightly clunky title, but I hope it's going to be interesting to you, even if you're not interested in soil or ecology or global change. But then, of course, I wonder why you're here. But I think it's hopefully still going to be of interest to you what I'm going to talk about today, because it's a bit more general. And so I'm going to start fairly broad. And this is like when we, in various human endeavors, there's often two equally good solutions to the same problem. Here in architecture, for example, it can be the Sagrada Familia, which is extremely complex and convoluted and complicated, or it can be equally excellent. This is the Neue Nationalgalerie in Berlin. Or in art, the same artist, even Gerhard Richter, probably Germany's most famous painter. There can be very, very complex paintings, and there can be super simple paintings. This is like Schwarz-Rot-Gold. In science, though, simple is always considered best. And that actually goes back to what many of you, I forgot to ask you if you've heard of Occam's razor before, but many people actually have not. This is the principle of parsimony. It goes back to William of Ockham, who's a monk that lived here in the 13th and 14th century, basically said it is futile to do with more what can be done with fewer, and plurality should not be posited without necessity. And that basically, even though if you don't know that principle, you probably apply it every day well, you do, if you do experiments, you apply it every day because you design typically the simplest possible experiments. And if you do observational studies or observational work and you pick a model, you always use certain criteria like AIC or BIC that basically are there to select the simplest model. If you add additional parameters, you get penalized for that. The idea really goes back to probably Aristotle and has really influenced the way we do science today, I think. What I did not know is that this is actually quite an active field of philosophy of science. I've attempted to read this book here. This is one of the books on Occam's razor, which is basically the philosophical look at this issue. And so the basic idea of this talk is that in the environmental sciences, but probably in all sciences, in all experimental sciences, this idea, Occam's razor, or should do simple experiments, has probably or very definitely led to only very few factors being considered in experiments at the same time in any one experiment. And I think this is, love to be corrected if this is not true from your vantage point, but I think this is true of certainly in all of environmental sciences and biology that I know. Even though we of course know that the world is much more complicated than that. And so I'm going to talk to you today about two case studies. One is the case of multiple global change factors. And if you don't know what global change is, I'll just explain that in another 2 slides. And then a very short part at the end on joint effects of multiple management practices. That is something that we've just started in the lab, whereas multiple global change factors has been going on for quite a while. So the first case study, this is basically the view we have from our institute building in Berlin, which is in the botanical garden. The building itself is slightly ugly and there's nothing close to this palace here, but we have at least a nice setting within Berlin. So soils are affected by multiple factors of global change. So first of all, what is global change? Came sort of as a shock to me about 2 years ago. I learned from science journalists that neither in Germany nor in the US nor anywhere else that you knew about, people in the street, or if you talk to anybody meeting in the supermarket, except maybe the Eon here on campus, but would know what global change is. People simply don't know. That word hasn't sort of entered into the common vocabulary, but people do know what climate change is. If you ask people, they've heard that German Klimawandel and climate change in German. So it's the changes in patterns, basically, of precipitation and temperature. That itself can be very complicated. I don't mean to imply this is simple, and it's also not exceedingly well understood. It's beginning to be quite well understood. But global change is so much more. It's basically all factors, including climate change, that are caused by human activity, directly linked to human activity, that are global in occurrence, and that affect biological organisms. Actually, if you want to find the definition, you'll be hard-pressed, because there basically is never a definition of that. It's always defined by enumeration. Global change is when you have climate change, elevated CO2, and so forth. I'll just showcase one factor here, which is one that actually most people in the lab work on. It's microplastic, the stuff that comes off your shirt. And this is a 2 millimeter soil aggregate. Soil aggregates will feature again a little bit later in the talk. I'll show you here. It's a little crumb that soil consists of, which is, I've spent like 25 years of my life so far studying. And these things that you see sticking out of this aggregate, those are your microplastic fibers. So they get actually incorporated into the very fabric of soil. It's not what I'm going to talk about here mostly, but it's just one of the factors that I'm going to mention. So if you look at global change in soil, or anywhere else for that matter, it's a problem of many factors. So the green ones are the sort of the big 4. Those are the ones that most work is done on. It's warming, drought. Those two are also commonly combined as climate change, as I mentioned, including flooding and things like that. Then you have nutrient enrichment. This is atmospheric nitrogen deposition. I don't know if you know that, but if you stand anywhere on Earth, there's always nitrogen falling right down from the atmosphere on you. If you're in Berlin, it's 40 kilograms per hectare per year. If you are in the Netherlands, it's 100 kilograms per hectare per year. So it's pretty close almost of what a farmer puts on the field. It just rains down of the atmosphere. And then you have, of course, elevated atmospheric carbon dioxide, which in addition to causing warming because of the enhanced greenhouse effects, it's itself also a factor of global change because it affects plants. Not so much soil, but of course, plants grow in soil, and so the effects on soil are therefore indirect. But I already mentioned microplastic. There's a lot of synthetic organic chemicals. There's 350,000 known chemicals that are used industrially in the world. So that is basically a bottomless pit. This is just an estimate. It's for sure way more. You have salinity, you have soil compaction, heavy metals, artificial light at night, invasive species, enhanced levels of antibiotic resistance genes. Those are all things that we work on. So the list is actually very long. The ... is basically the decisive thing here. So this is very much a high dimensionality problem. If you say you want to study global change, you actually ought to be concerned with all of these things. What does the reality look like? The reality looks like this. Very much not what it should maybe look like. This is a systematic mapping that we did a couple of years ago, where we just looked at a couple thousand papers that have been published on this topic of global change and soil from experiments. And we just counted the number of factors that were dealt with in any one paper. So this is the count of number of papers. These are the number of factors that were combined in that one study. So in the little gold color, you see most by far papers, and I think this is unsurprising to anybody, deals with one factor. This is, of course, important because we do experiments with one factor because we'd like to find out what it does and how it works and what the mechanisms are that underpin any effects. So nothing bad about that. But you see quite a sharp drop to 2 factors being considered. And there's almost nothing here like in the 3 and 4 and really nothing beyond that. But I've just shown you in the slide before that the list of factors that you ought to be looking at totally, but also in any one site, when you think about it for a moment, wherever you stand, there's going to be many, many of these factors influencing your soil and your vegetation. So this is hardly being studied at all. So 98% of all papers ever published on this topic, and we've since looked at like another 10,000 papers, and the pattern is still exactly the same. Looks at just 1 or 2 factors at the same time, which means we know really next to nothing about what happens with multiple factors, even though this is the reality of global change. So you could ask yourself, why is this work not being done? Came up with a few reasons. First of all, there is a psychological thing where people just think if you deal with the main factors, you basically got this thing covered. This is actually this signature of Occam's razor, if you will. There's this reliance on looking at just the main factor covering it all. Of course, there's logistical constraints. You have to gear up to studying all these various factors. Global change biology is a huge field now. There's a fair amount of research compartmentalization. So people that work on invasive species publish in different journals and go to different conferences than people that work on warming. And then, of course, the most pressing problem is, and why this is not being done, is the so-called combinatorial explosion problem, which means like for every new factor that you add in your experiment, you basically, if you have only 2 levels per factor, you double the number of your treatment combinations. So if you have 2 factors with 2 treatment combinations, you have with 2 levels, you have 4 treatment combinations. Let's say warming and drought with each on and off, that's 4. If you added another one, it would be 8, 16, 32. 64, 128, 256, 512. And if you had 10 factors, it would be 1,024 treatment combinations. And then you need to also still replicate that stuff. And so that is simply not possible in ecology. It is possible in some fields and has been done maybe once or twice. I've seen a 10-factor study being done with pipetting robots and E. coli. But if you have anything slightly more complicated than that, where the readout is not just OD, so optical density, then this doesn't work, basically. So this is not possible. And that's kind of a shame, because when you look at experiments in ecology and environmental sciences, and I'm sure it's the same if you're in another field, we have quite a diversity of the experiments that we do. We have different degrees of realism. So in my lab, we work with in vitro experiments on petri dishes, actually quite a lot. We do a lot of greenhouse and mesocosm experiments and field experiments that I'll show you a bit today. And there's also quite a bit of complexity in how we represent time. But in terms of factor dimensionalities, a number of factors, basically this cell here is empty, the high complexity cell, because simply there's no way to do it. Because this combinatorial explosion problem. So, and the next slide is basically the idea of this entire talk. So this is the one solution to this high dimensionality of factors that I came up with by stealing an experimental design from a related field, biodiversity ecosystem function. where people had also this problem of dealing with the number of things. Basically, this is a different question. The question that I'm asking here now is like, what if I had more and more factors? Rather than looking at all the factorial combinations, which we know is not possible, I can simply ask what happens if I have 1, 2, 5, 8, 10 factors along a series, a gradient of factor number. And it turns out there is a design in a completely different field, biodiversity ecosystem function, where this has been done. And this is by Dave Tillman, had a paper in Nature on that in 1992. Basically, what he did is the following, or his group did was the following. They had a pool of, in his case, species, plant species, and they created this gradient of like a control, which would be no plant species, one single plant species, and then maybe 2 plant species and many, many combinations of plant species. But there is one problem. You have to focus on the number of items, and you cannot confound the number of items with the identity and the composition of these items. So what he did was he just selected at random from this pool for every experimental unit, every like little experimental thing that you do. Let's say if the factor level is 2 or the species level is 2, 2 species at random from this pool. So then you have, let's say, 10 replicates of 2 species. Each of these replicates has, by chance alone, probably 2 different species, but what they have in common, it's 2. So you're asking a question about, what if I have 2 species? But I have to do this trick with the random sampling, because otherwise I confound composition and identity of the species with the number of 2 species. Does that make sense? So the idea here is simply, let's replace species with global change factors. So rather than having a bunch of species in here, we have global change factors in there, like the ones from the list that I showed you. And now let's say for factor level 2, we pick 2 of these global change factors at random for every experimental unit. This is a little tube of soil, the 30 grams of soil in it. I'll show you the next picture. So the first replicate of treatment level 2 maybe gets microplastic and drought. The second one maybe gets insecticide and warming. The third replicate maybe gets salinity and microplastic, and so on and so forth for 10 or however many replicates you can do. And then you do that also for 5, for 8, and for all 10 factors, just by random draw. Yeah, I thought this was a great idea, so I asked my people in my lab, who wants to do it? I have this amazing idea, and absolutely nobody wanted to do it because, of course, it seems like almost silly of an experiment because you're literally comparing apples and oranges, right? Because this is all kind of different treatments, and you're comparing them within the same treatment group. So what would the expectation would come out would just be garbage. So nobody wanted to do it. So I ended up setting this up myself. But then, of course, people felt sorry for me because I was like, where is the summons and how do I anyway, so after a while, people started also helping because I looked rather sad. So this is what part of this experiment looked like in a climate chamber. So we have these little tubes here. These are the experimental units. And to each of these experimental units, these factors were added at random, as I said, along this gradient of global change factors from 0, which is the control, to 1 factor at a time, for 10 factors replicated separately. That's actually the biggest part of the experiment. And then this gradient of global change factors. And these things are just temperature controllers because we independently administer temperature. Okay, so this is what comes out. Sorry for a busy slide, but you can ignore most of it, and I'll walk you through it bit by bit. So here we have our response variable. The thing that we measure is water-stable soil aggregates. Remember the scrum I showed you in the beginning, the soil aggregate? This is just how stable it is when you very gently tumble it in water from each one of those little tubes. This is our output variable for in this slide here. Here's our treatment. So here we have the control, also for easy identification. That's that dashed line going right across. This is when you do nothing. And then we have the 10 factors. So we have drought, nitrogen deposition, antibiotics, fungicide, copper salinity, insecticide, and so on and so forth, each separately replicated. And as you will notice here, some of them have slightly positive effects, 4 of them. 3 of them had negative effects, also the effects not huge. And then about another 3 of them had neutral effects. That's going to become important in just a second. Yeah, and this part is what I just explained to you with this gradient in global change factors going from 1 to 2, 5, 8, 10. This is the bit on random sampling. One factor is here. This is just simply all of these data points here, excluding the control, because it's also one factor. 2, 5, 8, and then all 10, there's only one way you can sample 10 out of all the 10, namely those 10. And, you know, without any of the fancy statistics here, what you can basically already see with your bare eyes, that is not random at all, what happens here. This is like a straight line going down in terms of this variable here. Even though about a third of the points were positive in their effects, third of the points negative, and third of the points neutral. So actually 70% of the treatments had neutral or positive effects. But if you combine them, the effect is very strongly negative. This is literally was the first thing that we measured because it's very, very easy, costs nothing, just nerves in a bit of time. Then, of course, we know what each of these data points is one of these little tubes of soil with a particular combination of factors. And we know what those combinations were, and we know what the factors did by themselves because we actually replicated them separately. So we could formulate expectations of what this combination of factors should actually look like in terms of the effect. This is sort of depicted here. So the null models, there's 3 commonly accepted ways on how you can combine effects. You can add them up, you can add the proportions up, or you can say the strongest effect wins. Those are the 3 null models that are commonly done in this kind of work. And you can see that these null model expectations deviate more and more, and for the last 2 levels significantly from the data that we actually measured, which means the null models do not explain the data. So something else is going on. Basically, the null models are under the assumption there are no interactions. So this means there are some interactions going on. What's astonishing is that just knowing the number of factors that are acting on that soil, which is something unbelievably crude when you think about it, you don't even know what factors. Is it warming or microplastic or insecticide? So just knowing the number of factors, we can explain over 50% of the variability in that experiment. Pretty astonishing. If you add more information, like you know which factors did you add, then of course this jumps up. Okay, the next slide looks exactly the same, and the one after that as well. This is a little bit different. This is what we measured here, soil water repellency. It's basically just how hydrophobic your soil is. So when you haven't watered your plants for a while, and then you add back water, then it pearls sort of off of the surface, and that is soil water repellency. It happens to form on soils with a bit of organic matter and sand. So these are sandy soils from Berlin, from the grassland, and so they have a propensity to show water repellency. But none of the factors, except for this effect on drought, which is what I just explained with your plant in your room, there are really no effects of any of these factors on that water drop penetration time. It's a wonderful, yeah, sure.
Effect size. So did you actually change the effect size as well?
No, this is the effect size of the single factor effects. We have only the effect size of the single factors. Yeah.
Thanks.
Right. So, I mean, except for this little blip here with the effect on drought, none of the other factors had any effect on water drop penetration time. Water drop penetration time is just simply the time in seconds until a tiny droplet of water that you very carefully place upon the soil surface has completely infiltrated into the soil. It's almost meditative in the lab. But look at what happens when you have an increasing number of factors. And so with 2 and 5, well, with 2, nothing that much happens, but from 5 factors on, remember this is where nobody looks, from that slide that I showed you a couple slides ago. It kind of breaks off at 4 factors. So from 5 factors on, and then also 8 and all 10 factors, there is this rather massive increase in water repellency. And that's not a small effect, though. So it's almost 40%. Some of them like went over a minute. Takes from immediately infiltrating into the soil, which is the control, water penetration time of like 0 seconds, which means it immediately goes in, to like it takes almost a minute for a droplet of water to infiltrate. This is a pretty important variable describing fluxes of water and hydrology. Yeah, and of course, your null models don't explain anything because there's not much information contained in here. So it's like a surprise. And actually, we don't know what causes it, neither does anybody else, because this is the tricky part. You can now not pick apart exactly which interactions cause this effect, because that design doesn't allow you to do that, because you have not replicated any one particular combination. You have only replicated the number of factors, but not any one of the combinations are replicated. Yeah.
Just a quick question now, but isn't it possible to do some kind of knockout experiment, like starting from 10 and then just remove 1 factor?
We have exactly done that. I cannot show it to you because it's not published yet, but it's we have done that. We've just gone the other way around, and then we have actually found that the effect of a single factor in size and direction can be the opposite of the effect of that factor when all the other 9 factors are there. So something that is, can't remember which way, when it is positive as a single factor, and this is the case that we found, it turned out to be negative in the combination together with all the other factors in these remove 1 experiments. We thought we were the first ones to do this remove 1 experiments, but it turns out we're not. Somebody's already done that in ecotoxicology. Yeah.
Can I just ask, was the soil sterile? Like, did you
No.
sterilize it?
No. They said everything in it. Yeah.
So how did you make sure it's always the same composition of microbiota there?
I'll show you the composition. This is in 2 slides, but basically we mix it well. That's it. Yeah, so we wanted all of the biodiversity in there because we also wanted to measure that, how it's affected. I'll show you that in just 2 slides. So I'll just quickly show you this one. We measured also, these I've already shown you here. We also measured decomposition rate and respiration. And we can look at these patterns. They are broadly similar. So for a bunch of these other variables, we see that same effect as well. And now for the diversity. So in this experiment, we just did fungi, because my lab mostly works on fungi. ASV richness is amplified sequence variant richness. You can think of this as something quite like, but not really, species. And I know this is a busy slide, but if you look here, there's a very nice decrease in the number of taxa that are in the soil. So they went extinct. They're no longer, we can no longer find them. So there's on average about like 40 species of fungi in each one of these tubes. And of course, there's some variability, but it's not very variable. So the mixing was fine. There's a super beautiful compositional change here that is really like pearls on a string. This is basically just from the ordination axis of composition. This is the first ordination axis, captures like first dimension of compositional data. And there is a directional change in composition, which is because, for those of you who like fungi, we lost basically the Basidium mycota disproportionately much compared to Ascomycota and Mucormycotina, the phyla that were in there. I said everything was in there. Dispersion is distance from the centroid. And so basically we see for also biodiversity data this very clear pattern of directional linear change, basically. It's completely unexpected. Yeah, so you might think, yeah, well, this is just one soil and as one particular group of 10 factors, but we've since repeated this like very many times. This is one of the repetitions, which is a much bigger experiment by a much younger person. And they used a different soil and also used different factors. and combinations of factors as a consequence of that, and found the same pattern. In addition, in that same experiment, this was factorially crossed with low and high biodiversity. So we basically did exactly the same experiment with low soil biodiversity using dilution to extinction, basically, commonly used approach in soil, and then high biodiversity. And the y-axis is simply the difference between low and high biodiversity across this number of factor gradient. And all of these wild little squiggly things and colors are different response variables that we measured. You don't need to concern yourself with those. But you see that there is a biodiversity effect at factor number 0, the control. This is what we know very well. A more diverse soil has a higher rate of processes that it supports and properties that it creates. And as you increase the number of factors, you can see this difference between low and high biodiversity dissipates, which means one way to look at it is you can say that high biodiversity is not going to help you. You know, if you have that many more factors active, then also in part because you lose biodiversity, which we've documented here also for fungi, protozoa, and other groups, not just for bacteria, fungi, protozoa, not just for fungi. So you lose diversity because of the high diversity that you start with is not going to buffer that level of impact with this many factors. So in the end, there is no difference anymore between low and high biodiversity in all these process rates and properties. We did another experiment that just came out by a graduate student in the lab, and what they have done is actually 4 PhD students working together on this project, which we never had before. They made a pact coming in and said, like, we are going to do each our own PhD, but every year we're going to do one thing together. Super cool. So they did that the entire PhD, and this was the first one. So what they did here is that they had a factor pool of 12 factors, so listed here. And they did 2, 5, and 8 factor combinations. But here the question was, what about the dissimilarity of factors in any one experimental unit, in any one of these soil tubes? So you add like 2 or 5 or 8 factors, but for each of the combinations of factors that you add, what is the role of how dissimilar these factors are? Dissimilarity defined as the effect. So it's effect dissimilarity. And so you could basically ask, or we asked, is it worse for the soil and its biodiversity and its process rates if you add 2 factors that are very dissimilar in their effect? Or is it worse if you add 2 things that are relatively more similar in their effect? Because you could think that they basically hammer on the same point to the point that it breaks. Or is it worse if if the damage is sort of spread out across various process rates and components? And in order to do that, I'm not going to show you the data because it's super complicated to explain, but basically they had 50 units for each of those. So they did 150 different communities of global change factor combinations. I know of no experiment that has done that before. And so they could basically create a dissimilarity gradient within each level of factor number, and then with 2,500 lines of code in R, show that it is really the dissimilarity that explains quite a bit of additional variability in the data set beyond the number of factors. And that the reason for that is that you have synergistic interactions that are becoming more prominent when you have greater dissimilarity of the factors that affect any one soil. So this is a rather complicated thing to do, but we had to do that because there is no other way to get at how do factors interact, because this design doesn't really allow you to do that as random sampling. You have to get at it from another direction. So this was our attempt to find out more about what is it about the factors that are causing these effects. Are they more dissimilar or more dissimilar when they have, or more similar when they have worse impacts? Now you could say, of course, these are all like these little soil tubes, and they're a little bit weird. And in reality, soil is not in these little tubes, but it's out there. So we set up a field experiment that was just finished. It ran for 3 years. We are now actually going to run it on, but in a much reduced effort. So this is exactly the same design as I've just shown you in these little tubes, but now in the field in 1 by 1 meter about grassland plots that was sown grassland. What you can see here, those little things here are the roofs. So those are the rainout shelters that intercept about 40% of the incoming precipitation. This is just 10 minutes walk from my institute in Berlin. And these things here are open top chambers. So they are passive warming chambers, which raise the temperature by about 1.5 degrees Celsius. The other 8 treatments you can't see because they were added to soil. There were microplastic, salinity, copper, pesticides, all these various things that we already mentioned. This ran 3 years. It's, as I said, exactly the same design. Looks like this. And there were very clear effects, not on soil, annoyingly enough. I've talked to some other people that have done big experiments and they see the effects on soil after 4 years for reasons nobody knows. So this is why we are now running it for a little bit longer. We saw in the last year, we saw effects also on soil processes. But we see exceedingly clear effects on plant biomass. I mean, a straight line down, just like what I've shown you with these other data. And extreme effects on diversity because we lost a lot of species. The more factors were basically active. And also our favorite root symbiosis, avascular mycorrhizal fungi were also suffering, also a linear decrease along this factor number gradient. So that makes me think this is probably real because we've also seen it in the field. This is usually the litmus test because, you know, this is much more complicated. All kinds of things go on in this field. It's very variable. This is not well mixed. Like, I asked the question before about how could you make sure this was the same community every time? We didn't. You know, we know how variable it is. It is super variable here, but even given that variability, there were still these effects.
Just a quick question.
Yeah.
Is there some kind of spatial interaction kind of effect?
Yeah, excellent question. So we set this up ourselves, including me, and I hurt my knees because what we did is these are 156 plots, and each one of those plots has like 30 centimeter deep barriers around every plot. So they were basically hydrologically isolated from the adjacent plot because there's water running off of the roofs and it drips down right here. And if there were no barrier, it would just flow right back in and you would have no effect there. Yeah, so we had to do that, but we can't just do it for the rainout shelters because this is disturbance. So we had to do it for everything. So it was a tremendous pain. Yeah. Actually, this was just done on institutional operational funding. It's one of the nice things to be in Germany or at OIST because you can just set stuff up and not worry about funding. Yeah, but then, of course, we also wanted to know, do we see this effect in observational data? So I've talked so far only about experimental data, and this is the focus of this talk. Well, I'm going to show you one slide about observational data, which is when you go out there on our planet and you sample a couple hundred different ecosystems, you measure things, you don't interfere in the normal course of events as an experimenter by applying a treatment, but you just capture the reality as it unfolds out there. So we did that here. This is about 200 sites from different surveys. Now, this is continuously measured data of various pollutants and other global change factors. And so you have to now transfer the continuous data into basically counts of factors, because this is what you have on the x-axis, the number of factors. So there's two things you have to do. First of all, you have to define a threshold. When crossing that threshold, you count that factor as present or on. And when it's underneath the threshold, you say it's not there. But then, of course, what threshold do you use? It's going to be totally arbitrary. So the answer is use every threshold. So this is what you see here from the dark purple to the green and yellow. This is a higher and higher threshold in terms of the percentile of the entire data set collected on Earth. And you can see that the higher the threshold, so the higher the barrier to counting a factor as on. So basically, this is a higher intensity of factors that needs to be reached. the more clearly negative that relationship becomes with increasing number of factors active. Now, you might be surprised why the counts here are so low, because there were quite a lot of factors measured or listed here. The reason for that is we had to group factors together to avoid multicollinearity problems, which were rather massive in this data set, as some variables tend to co-vary, like many heavy metals usually are like that. So this is why there's fewer factors along the x-axis. But it's very nice to see, also in observational data set, where this is not an experimental artifact now, because this is just observed. Of course, then you don't have mechanistic resolution, anything like this. We could see that also all of the models selected the 75th percentile, so the high factor level, and the multi-stressor variable explained not a huge, but a distinct proportion of the variability if you forced all of the individual factors in the model already. So this basically captures the higher level interactions in the data set, if you will. This is that first part. The second part is very, very short, so don't worry. I know it's already gone for 40 minutes. So basically, the summary of that part is both factor number and factor dissimilarity seem to be important for explaining results of soils when they're exposed to these many global change factors. Now, this is usually where I ended my talk, and it's a little depressing because people think, you know, it's already bad, but it's actually, I've just been telling you, it's far worse than you think it is, most likely. So it's basically a depressing method. So we thought about, could we take that same idea of sampling things from a pool of factors and use it for good, basically? And so the question we asked is, can we restore soils better? Can we unlock benefits in restoration that are much better than what we could achieve before by using multiple management interventions now rather than global change factors, so something good? I'm just going to basically show you the idea. This is, by the way, taking stock of the experimental work done from a meta-analysis on restoration ecology. You're looking at many, many hundred papers. Does that look familiar? I mean, this is a completely different data set, a completely different set of ecology, and of course, you see exactly that same pattern, you know, that Occam's razor signature. We only look at few factors at a time. So the idea is now, can we combine increasing numbers of management interventions, do this in regional centers, because usually, as always, there's only one or two factors experimentally examined, and can we get bigger effects? I'm only going to show you one small data slide here, and you can only look at this here. So this is the amendment diversity. We have a couple more papers out now, but basically these are the number of factors and management interventions, in this case, additives, carbon-containing additives are listed here. These are the individual effects. Basically, you don't do much. If you add one or if you add two, also no effect. This is plant biomass. But if you add 5 factors, then we have a fairly huge effect here that we cannot explain yet. And we don't think it's just nutrient addition effects, because you would have expected some effects down here as well. So we have done a couple more experiments now. The effects are not huge, but what we have found, and there's also just a paper by Richard Barchett's group that came out that used exactly this idea as well, that what seems to be positively affected if you have more and more factors, management practices, is ecosystem multifunctionality. That means if you have individual factors, maybe you can have a benefit for certain specific functions, but they're trade-offs with other functions. Is actually what we observed in our latest study as well. But if you do many, many factors at the same time, then these trade-offs appear to disappear. But nobody really knows why yet. But it seems like overall the benefit for the ecosystem, and as measured in ecosystem multifunctionality, that becomes more solid the more management practices you do. We just submitted a paper on agricultural practices and we see exactly the same pattern as well. Of course, when it comes to these applied things, and there's still much more work going on in our lab now, then of course you have to also ask yourself about cost. But this is not something that we have included yet, because if you have more factors, maybe then it's also more expensive to do. And the latest thing in the last slide I'm going to show you here is that maybe this has also consequences for conservation, which is not really my expertise. But one major concept is climate change refugia, where you basically offer species places to retreat to when the climate warms. But that mostly ignores the impact of other global change factors. Just explained to you one of the first slides. This is a multi-factor problem. So maybe we need to move to defining and designing global change refugia, where either one particular factor is at a low concentration. There are like dark night refugia already, where there's no light pollution. That's one example. But maybe we need microplastic refugia or low antibiotic resistance gene abundance refugia. And ideally, global change refugia have low abundance of all those global change factors. And with that, I just want to conclude with these take-home messages. So I think hopefully could convince you that global change is really a multifaceted problem, and that we have at any one moment many problems exerting themselves on any soil and ecosystem. We found this clear signal of the number of factors at a variety of different levels of complexity that I explained to you. We might turn this around and harness the same approach of sampling from a pool to unlock benefits of higher levels of management practices to have better restoration success. And maybe overcoming Occam's razor thinking may be important going forward in the environmental sciences, and maybe have other ideas on how you could really attack this problem of multiple factors, for which there is nothing in your experimental design book right now. And I wonder if this is a similar problem in other parts of the sciences. And with that, I'd like to thank my lab. And major funding for this came from ERC Advanced Grant, and yeah, also these various other programs, and the Bundesministerium für Bildung und Forschung. And thank you for your attention.
Thanks so much, Matthias. So we have time for some questions. Any questions? Yes.
Fantastic talk. Can you go back to before the second part, when you were talking about moving from lab experiment to observational data? Yeah, so there's something which confuses me about the curves in the top plot.
Yeah.
So before, when you showed the same plots for experiments,
Yes.
the curves look quite non-linear, which is what I would expected.
They look quite linear.
Here they look linear. Before they look non-linear. Here they look quite linear, which is
Quite linear, and actually linear model is like the best fit. I mean, all of the experiments,
now
it was linear. The one exception maybe this year, where we don't know what's going on with water repellency. But yeah, I mean, basically, if your question is about linearity or non-linearity, I think with our experimental setup with having basically 0, 1, 2, 3, 5, 8, 10, we have like 6 or 7 data points. I think we cannot really detect deviations from linearity with any good confidence. Yeah.
Yeah, kind of try to reconcile. I would have said that, you know, there's a parity effect, then it should be non-linear, because you would have linearity if these things just you know, 2 effects is twice 1 effect. And so yeah, I try to wrap my head around the fact that it's almost linear.
This is a discussion we also had. Yeah, yeah. I also have no answer to that.
Okay.
Yeah, but I think the real answer is it may be non-linear.
Not linear, it just looks
We
linear
just
because of the error.
it just looks linear because we have so few data points, basically. Yeah.
Okay.
So the idea is that to do the next experiment would be using maybe 30 or 50 factors. That wouldn't be really very difficult, I say here, because somebody else is going to do that, hopefully. But the problem will be to replicate the single factors. Really doing the combination is not going to be tremendously more work than what we do now already. But then we could have really an extended gradient. And with an extended gradient, we could look at non-linearities like breakpoints. I was personally surprised at the whole thing anyway. I thought this would be just chaotic, it would just be idiosyncratic noise. And so did everybody else. But it would be really interesting to see breakpoints, sort of tipping points or whatever you call them. And yeah, we have no power the statistical power to detect any tipping points now. Yeah.
Thanks.
Thank you. That's very nice. The question maybe in a similar direction is the question if there's anything that one can understand, right? Because there are so many factors now that either they're all sort of interacting non-linearly, that then it's very, very difficult to understand what's going on, or there's potentially a small, sparse set of interactions of things that actually really kill you, right? That really change something. Did you observe sort of like structured or sparse deviations from the linear null model? Because you would expect the linear null model if things don't interact. And the question is, are there just some combinations that drive the overall mean effect that you see, or is it just that everything is stronger if you combine many things?
Yeah, we can't do that because we have not replicated any of the combinations. Huh? So for everything, we just have one data point. For like microplastic and warming, we have one data point. And so microplastic and warming may occur again in the higher factor combinations, but then with a totally random combination of other factors. So basically, any way we looked at it, we couldn't tease that out if there were key interactions. You can do that. I think there's like machine learning algorithms that tease out such interactions, but you need a lot more data points. That entire experiment was just 140 data points. I think you cannot get that out. Yeah. And even with the new study from Mohan and Hu Ying, there was 150 communities, and then add the single factors. This is another maybe 100. They're like 300, 150. I don't think this is the level where machine learning can pick these out. This is what machine learning people tell me. Yeah, I of course don't know how to do that.
Thanks.
But that would, of course, be amazing if we could pinpoint what causes this. This is like what also annoys all reviewers that we cannot say.
And how does that play when you intensify one factor? For instance, here you're picking, I add microplastic and warming, but to that level. And if you are increasing, you're doubling or like 10 times the amount of microplastic that you're inserting in your sample, how does that compare with playing with multiple factors? Is it also linear growth or?
Right. I mean, I get this question every time. It seems like we should actually do this experiment at some point, but we cannot like cross this design with also having different intensities that would blow up. So that's not possible, but we could have individual intensities increased and compare it to the multiple factors. This is what you mean, because I've heard this question before. It's a very good idea actually for our next experiment. The thing is that the levels that we chose for our factors were not totally sort of arbitrary. We tried to have these levels informed by things that we actually measure somewhere in Central Europe, let's say. But these are realistic occurrences of these factors. Like, I mean, if you take this idea and say like you have 10 factors, let's do 10 times the warming. We had 3 degrees or 5 degrees warming in some of the experiments. It doesn't make sense to have like 50 degrees warming, right? Because you would cook that stuff. Yeah. You would have to have a dose response curve. But for some of these, it simply doesn't make sense, right? I mean, it's not applied research in that sense, but we're trying to be oriented to what's actually occurring out there. So I understand what you mean, and I think we're going to do that experiment. Yeah, but I have no answer right now. Yeah. Actually, those response relationships are super rare. I don't know if it's different in your field, but hardly anybody does this with dose response. I mean, in a way, this entire thing is a dose response. It's just the dose is the number of factors, right? But if for any one factor, dose response curves are super rare. Like for microplastic, I know one paper that did actually dose response. And then they did analyze it with ANOVA, not with regression. This is like dose response is like a big blind spot. Yeah. Yeah, sorry, that's all I have now.
Any other questions? I have one really quick. So in that global meta-analysis you carried out on these, I mean, what was the distribution of your samples globally? Do we know anything about, you know, whether those trends differ, say, in tropical soils and rainforest soils and desert soils versus what are usually measured in like temperate European and American soils?
I think we did not have enough replication for each one of those because those were, you know, put together from different surveys that measured different things. So I think to drill deeper into the data into ecosystem types was not possible, but it would have been nice to do. Yeah.
Yeah, it would be really interesting to know just how resistant different areas of the world are to particular stressors, because that sort of harkens back to maybe the adaptability of the system and what it's experienced in the past.
Yeah, no, I agree. I think it's an excellent idea, but I think that there's limited number of surveys that measure a whole lot of things.
Yeah,
so I think that these surveys were all done by basically one lab where they measured a bunch of things on the same sample. You know, I mean, maybe what you could do is like you could collate a bunch of different surveys of more single factors with, you know, extrapolation to other data points where they haven't sampled. I suspect the uncertainty would blow up very, very quickly because you couple together too many different things from too many different surveys. But so this is why we focused on surveys where they actually did measure all these things on exactly the same sample. Yeah.
It's like we have to expand the nutrient nets kinds of globally distributed experiments and figure some more stressors out that way.
Right. I mean, this is also an idea we had. So we have there's an experiment like this in China, and then there were a couple of people that were interested in setting them up, and then they thought more about it, then they gave up. Because I mean, if you think about it, it is a tremendous pain in the ass to set this up.
Imagine all the emails that need to be sent. Cool. Any other questions? Okay, well, please join me again in thanking Matthias. And how much longer are you going to be here?
We'll be here until the 22nd of January. 22nd of June.
Got you for another year. 8 months. Okay, great. So please send him an email. He's very open, I assume, with your schedule.
Yes.
So happy to meet people across all disciplines.
Yes, definitely.
So thanks so much.
Thank you.